T-lymphocytes, T-cells. Production, activation, types and functions. Introduction. T-lymphocytes, commonly known as T-cells, are a vital component of the adaptive immune system. They play a central role in cell-mediated immunity, which protects the body against viruses, intracellular bacteria, cancer cells, and foreign tissues. Unlike B lymphocytes, T-cells do not produce antibodies. Instead, they directly kill infected cells or regulate immune responses. The letter T in T-lymphocytes refers to the thymus, the organ where these cells mature. Production of T-lymphocytes. Origin in bone marrow. T-lymphocytes originate from hematopoietic stem cells, HSCs, in the bone marrow. These stem cells differentiate into lymphoid progenitor cells. Immature T-cell precursors, called pro-T cells, are released into the bloodstream. Migration to the thymus. Pro-T cells migrate from the bone marrow to the thymus gland, located above the heart. The thymus provides a specialized environment essential for T-cell maturation and selection. Maturation of T-lymphocytes in the thymus. T-cell maturation occurs in two main stages. A. T-cell receptor, TCR, development. Immature T-cells begin to express T-cell receptors, TCRs, on their surface. Each T-cell develops a unique TCR, allowing it to recognize a specific antigen. B. Thymic selection. To ensure proper immune function and prevent autoimmunity, T-cells undergo two selection processes. Positive selection. Occurs in the thymic cortex. T-cells must recognize self-MHC molecules. Cells that fail to recognize MHC undergo apoptosis, programmed cell death. Negative selection. Occurs in the thymic medulla. T-cells that bind strongly to self-antigens are destroyed. This prevents autoimmune diseases. Only 2 to 5% of developing T-cells survive and become mature naive T-cells. Types of T-lymphocytes. After maturation, T-cells differentiate into several functional types. A. Helper T-cells, CD4. Express the CD4 surface marker. Recognize antigens presented by MHC class II molecules. Secrete cytokines that activate. B cells. Cytotoxic T-cells. Macrophages. Coordinate and regulate immune responses. Cytotoxic T-cells, CD8. Express the CD8 surface marker. Recognize antigens presented by MHC class I molecules. Directly kill infected or cancerous cells by releasing. Perforin, creates pores. Granzymes, induce apoptosis. Regulatory T-cells, TREGs. Suppress immune responses. Prevent autoimmune reactions. Maintain immune tolerance. Memory T-cells. Long-lived cells. Provide faster and stronger immune responses upon re-exposure to the same antigen. Activation of T-lymphocytes. Antigen presentation. T-cells are activated in secondary lymphoid organs such as lymph nodes, spleen. Antigens are presented by antigen-presenting cells, APCs, like dendritic cells, macrophages, B cells. Two-signal model of activation. Signal 1, antigen recognition. TCR binds to antigen MHC complex. CD4 binds MHC2, helper T cells. CD8 binds MHCI, cytotoxic T cells. Signal 2, co-stimulation. Provided by co-stimulatory molecules, e.g., CD28 on T cell and B7 on APC. Without this signal, the T cell becomes anergic, inactive. Clonal expansion and differentiation. Once activated, T cells undergo rapid mitotic division, known as clonal expansion. All daughter cells are specific to the same antigen. Cells differentiate into effector T cells, memory T cells, effector functions of T-lymphocytes, helper T cells, release cytokines such as interleukins, interferons, activate other immune cells, regulate antibody production, cytotoxic T cells, bind directly to infected cells, induce apoptosis without damaging nearby healthy cells. Role of T-lymphocytes in immunity. Defense against viral infections. Elimination of tumor cells. Transplant rejection. Long-term immune memory.